Good morning to everyone. First, I would like to thank the organizer for organizing this interesting meeting and also for inviting me to show you some results, perhaps of the first kit based on 6-die technology, and also perhaps one of the last generation of kits for capillary electrophoresis. But let me show you why we were involved in these studies. In 2011, we proposed to the ISEC program a proposal to validate a single 21 STR typing system based on 6-die capillary electrophoresis detection to improve some DNA data exchange locally and globally. The problem we have in Spain is that with the implementation of the new standard set of STR, the new European STR using this kind of kit like NGM Select or SX17, this will be inefficient comparison with our historical database, which is based mainly on identifier. So this is a local problem, but as you can see also in the table, in columns 1 and 2, we have also an historical and perhaps a bigger problem, which is the lack of compatibility between the European and the CODIS standard. So this could an efficient future exchange between these two databases. So trying to solve this problem, we propose this set of markers, mainly where all the STR included in the European and the CODIS standard set, and also these 3 loci that are also incorporating many national databases, D2, D19, and EC33. What we demand to the companies, apart from the set, was, well, it could be nice if we can have a good concordance of primer sequence design with Biosystems, and also to develop master mix with high tolerance to PCR inhibitor for case war application. This project that was called iDNADEX, Improving DNA Data Exchange, was finished in October the year, and during this project we have analyzed 2 types of prototypes. The one that I'm going to show you now that has been published by our group in FSI Genetics, and also another kit by Promega now is called the Fusion 6 color system. This slide shows you the general characteristics of autosomal STR markers included in the GlobalFiler kit that you all know very well, and also some forensic biostatistical parameters in the Spanish population. And just 2 comments of this slide. One is that it's a perfect concordance with primer sequences for all markers except for TPOX. So this is very good for concordant studies. And second one, that we have around 12 markers under 250 base pairs, so it's very good also for DNA degradation. And if we make some calculations in the Spanish population, you see that we have a better probability of identity with GlobalFiler compared with previous kits like NGM Select. It's 5 order of magnitude for the probability of identity, and more or less 2 order of magnitude when we calculate the probability of paternity exclusion in the Spanish population. As you all may know, apart from the autosomal STR, the kit also includes 2 new YSTR, which are really interested to reinforce the possibility of new alleles abdiamelogenin. So these are the Windel, very good for degradation because it's a very small target, and also the DIS 391. This is the list of validation parameters and the samples that we use in each study. Don't worry, because today I'm just going to focus on analytical threshold and sensitivity and stochastic threshold, so you can have an idea how the result compare with previous kit. And then I will center or focus the presentation on some experiment with DNA mixtures, DNA mixture of DNA extracts, and also DNA mixtures from different body fluids from proficiency samples or proficiency samples from proficiency exercise. I will show you also some species specificity studies and some stability studies with case type or case word samples. These are the methods that we use for DNA extraction, DNA quantification, PCR amplification, and capillary electrophoresis detection. I haven't time to get into details. These are the standard conditions, but just to recall something that is very important in the validation studies is the DNA input, the annealing temperature, the number of cycles, and the injection parameters are very important and affect all the threshold that we analyze in all these validation studies. So my recommendation is, if you made some change in these parameters, do and document the validation studies. We obviously use the GlobalFiler user guide and GMapper IDX, and also for comparison and concordance, we use some modules that we have in our LIMS system for comparison purposes. This is the appearance of a GlobalFiler profile. In this case, is from a reduced reaction volume of 12 microliters. In the comparison we have made in different saliva extracts, side by side, you can see that the performance is very good, even more concentrate in the case of the reduced reaction volume in comparison with the standard 25 microliters reaction. And this depending on the calculation you made, we can get 50 RFUs or analytical threshold, or in the case that we use the more, let's say, conservative, 2 times the difference between the maximum and the minor peak. We have an analytical threshold of 1. Evaluation of this kind of analytical threshold with the negative control that are associated with case work. Obviously, the analysis of this negative control are especially indicate to monitorize contamination, but also to analyze this kind of threshold. increase with decreasing DNA input. And regarding the stochastic or reference value for the stochastic, the value of the highest surviving false homozygous peak was in this series around 900 RFU for the market to 12 at 21 picogram. Of low-level DNA samples using the new collection of software that use this kind of parameters, the probability of dropout. With this kind of sensitivity study, we also look for the incidence of drop-in a low DNA input. And when I say drop-in, unspecific amplification of DNA from non-human species. We have made an interesting study against more than 20 genomic DNA samples from several human-associated microbial species, and in all case, we have negative results, so this is a coupling by GMAP or IDX. Regarding the minimum number of contributors, you say with 2 contributors we have very good results. The total number of alleles and the number of STR with 3 or 4 alleles are very good to differentiate and to is really like in the 2 contributors, and in this case only 2 STR are with 5 alleles. So be careful with complex mixtures, and with more complex of 4 and 5 contributors, this limit